Perfect. Thank you for the introduction, Jade. So I'm going to be doing Romer's Gap Palynology, a fossil sneeze to remember. So I'm primary author on this, but there's a lot more that goes into this overall project than just what I'm doing in the palynology. There's structural, there's mapping, there's other fossils. So like always, science doesn't happen alone. But what we're going to be looking at more specifically is how old are rocks and how do we age date them? So there's two different main methods. One is absolute or radiometric dating, and the other is relative dating. Radiometric dating, we're looking at the elements that are trapped inside minerals and looking at the rate of decay over time. The clock starts for these types of rate of decay or these elements in rate of decay when you have either extrusion of ash or basically magma starting to solidify. And you get these minerals crystallizing and cooling, and at that point you have the clock basically starting. However, finding ash layers or different types of volcanic layers can be very, very difficult sometimes, especially in the Carboniferous. These are just some of the minerals basically as they cool, how they would look either in an ash or in a magma. What we're going to be looking at though is relative dating. And that's really looking at the layers of rock and which ones are older and then which ones are younger on top. So we think of time as very linear. You can see that in this diagram, you have layers of sediment that are being deposited with the youngest ones or the most recent deposits on top and the oldest at the bottom. A great place to see this type of sedimentary structure or this very simplistic layering of sediment is actually in the Grand Canyon. And just to give you an understanding, there's about 100, that's just 150, or sorry, 1.5 kilometers, just for scale. However, rocks aren't always laid one by one, or sediments aren't always laid perfectly flat. You also have other things that are happening as these rocks solidify. And it basically changes the order of operations. So you might have things that are being layered one by one, and then they're broken through time and replaced. So these are the laws of superposition by James Hutton. And you can see this is one site in Scotland where he really discussed it further, where you don't just get layers that are being deposited flat, they're actually being tilted and then have cross-cutting relationships. In the same time, we had the map that changed the world. This was done by Sir William Smith. And, oh, excuse the grandfather clock in the background for a second. But this is the map that changed the world. And we're looking at basically what happened was he mapped Britain and he realized that these layers of rock aren't always flat. Also that he was finding different layers with different fossils in each. So if we look at a more zoomed in version of this map, you can see that from a cross section of all these different beautiful colors, they're representing different layers of rock. In a simplified cross section, you see that we have trilobites in some of the layers and whales that don't, and then dinosaur bones that are also being seen at the surface, but they're part of different layers of rock. They're shallowly dipping. And what we're looking at here is basically fossil succession. Seeing these fossils in different locations and we can start basically subdividing time that way. And we can use this to start correlating different layers of rock more regionally, like William Smith did in the map that changed the world. So more locally, Sir William Dawson and Sir Charles Lyell did this at Joggins. And Joggins was a proving ground for a lot of these big ideas in geologies, with layers on the bottom are older than layers of ones on top. And this was revolutionary in the 1840s. So if we look at cliffs of Joggins, maybe not initially a very good picture, but we can start picking out is that we have these very defined layers, again, slightly tilted. And we can also start picking out is the trees within these layers. And you can see that the trees are in different horizons. So there's a nice one there, there, and there. These basically hollow cylinders that are usually filled with goodies as bones and charcoal and gastropods, and then can tell us something about what's going on. But this is fossil succession, basically one layer of rock having a fossil forest on another layer of rock. So this is Lyell's, this is just an example of that in a laid out in Lyell's section of Joggins. As you can see that the fossil trees have different infill than what's around it. Another way of looking at this and basically subdividing time using these types of techniques in fossils is kind of the history of technology. Everyone has a phone, but they used to have tails. And if we start looking at time in this way, where we start seeing, we can use different objects or different animals and plants to basically start subdividing time, we can understand where we are a little better. If we get into more detail and we decide that something like an Apple iPhone, or the morphology changes of like something like an Apple iPhone through time, we can actually see, we can actually subdivide time even further or in a finer resolution. So this is what a lot of my project is doing, is basically subdividing time to a finer resolution. So when we think about that, what are the challenges in the macrofossil record? Well, they're often big, and fragments of skeletons, we have whole groups named from single bones, but also from a distribution point of view, they're scavenged, they're disarticulated, broken up, and they get decay. So, and we also have big gaps basically between these different evolutions, or what we can, I would say, attribute well between macrofossils. So what makes a good index fossil? So what are we really looking for to get this very fine resolution of age? We want something that's widespread. We want something that's abundant, short-lived, evolves quickly, and stays mostly intact. The reason is because we actually want to use these and the changes of these through time to create something like a range chart. So in this case, we're looking to find the age of an unknown fossil and using the fossils around it to put it into context, and the beginning and the end of basically our first occurrences and the last occurrence of those fossils to put into context. In the Mississippian period, so what age group I'm looking at, oceans are optimal. This is what the Mississippian would have looked like about 350 million years ago. And we would have been located in the tropical zone, so basically around the equator in this. But again, oceans are optimal. So what are we usually looking for? Something small, sand grain size, either shells or also single-celled organisms such as dinoflagellates or teeth from conodont, these very tiny worms that have these very detailed teeth, and these are on the micron scale. So the land equivalent is what I look at, palynology. So the stuff that makes you sneeze. It translates to the study of dust. On land, this is pollen and spores. Pollen and spores are often carried or often associated with bumblebees for pollination, but they're very small and they can stick on basically anything, and they're abundant. They can also be carried by wind, and they can be carried very far. These are much smaller particles than sand, and they can make it all the way across the ocean. This is a sandstorm that actually happened in 2020 and blew Sahara Desert winds all the way over to Florida. So here's an example, a nice bright sunny day in Florida. It's pretty snowy here, so it would be much nicer, but you can see how those particles have really clouded the sky after that dust storm kind of came through. So just how widespread these things can really go is quite amazing. They're also transported and trapped in aquatic environments, as you have spores, which tend to be a lot thicker or thicker walled, tend to be transported by water. And what you're looking at is basically these things settling out as water movement becomes slower. So what are we looking for? We're really looking at the change of these different spores through time, and we're using those small changes in spores and pollen to subdivide time. Tiny fossils have a better chance of being preserved and have a more complete record through time, not just of fossils, of spores, but also of other basically single-celled organisms in the marine realm, but also pollen and a couple other important architects and acrotarchs, as well as other different microorganisms. So pollen and spores can be used as a proxy for plants, just because there's so many of them. If you look here, you can see that there's like tiny pieces of pollen on the anthers of this plant. But then if you kind of look even more closely, there's tons, there's lots. These are just SEM images of more modern day pollen, but we can do this for almost any type, and it's a really cool process. So if we start looking at just even how many thousands are on one flower, and we start looking at fields of flowers and then fields of flowers over the whole earth, you're looking at billions and trillions of pollen grains per year. And just because you have pollen and spores doesn't mean that all of them are going to be flowering or they're going to eventually become a plant. A lot of them get stuck in your sediment and never be anything more than a grain of sand. Now, if we look at just even that's just 1 year, over 400 million years, you can kind of understand that these things are abundant, well spread, and well distributed. But spores have to come from somewhere, and they come from plants. Some of our earliest understanding of plant comes from the Ordovician, Cooksonia, these early vascular plants that do produce spores, but they're very small. That's a 7 centimeters just for scale in that diagram. And then we start getting into, so they're very small and shrubby as we kind of are really like about 400 million years ago. And then we start getting into the Devonian, where we have shrubby forests of the middle Devonian, getting into what you'd think of like standard larger forest trees and different things near the late Devonian. As we transition into the Carboniferous, though, we start getting these really lush green forests with trees, calamites, all of these wonderful things. So very much if you think Joggins, or if you've known anything about Joggins, is that it's these rainforest wet type of environments a lot of the time. And also beautiful, beautiful ferns. So spores are known to come from ferns, and they are abundant in this time period. As you move further or into younger rocks, into the Mesozoic, you start getting also a change into true woody plants, as well as into some of our first flowering structures start in the Cretaceous about 130 million years ago. So your modern day examples of basically anything perennials and annuals, anything that kind of looks like a flower, really date back to only 130 million years ago, whereas what we're going to be looking at is 350 million years ago. So just to give you kind of a time frame as to where we are. What's important about this time frame, though, is that we are looking at this about 360 million years ago. It's this transition from the Devonian to the Carboniferous, and the evolution of tetrapods happens at this time, just as you have these lush tropical rainforests basically starting to build. So at this time, we have Roemer's Gap, and this was originally recognized by Dr. Alfred Roemer, and he realized that there was something wrong. We had the fish of the Devonian, and we had these very what's the word? We have the fish of the Devonian, basically very well established advanced vertebrates by the time we get to the early Pennsylvanian, places like Joggins, but there is nothing really in between, so we weren't really sure what happened. So again, we had these Devonian grade tetrapods. So their fins are not quite all together as far as having 5 distinct digits yet. And then we have this gap into basically the early Pennsylvanian, where we have the amniotes. So things that can lay eggs on land, and basically advanced reptiles. This gap also extended to the invertebrate realm. Basically, we're not seeing that diversity that we should be seeing in the vertebrates or the invertebrates. But realistically, this comes down to some of the work that Matt Simpson's doing as well on the footprints in the area, is that this may not actually be a gap. What might actually be happening is that it doesn't exist, and this is a research gap. And this is being filled in by work that's being done in Scotland, work that's being done by Matt Stimson in New Brunswick, and then also reevaluation of work that's being done in Nova Scotia. So this all kind of came about though, because we discovered footprints and bones in New Brunswick, and we needed to understand the context of them. So understanding the relative age was important because it shows us that basically where we are in tetrapod evolution. So the three big sites that Romer's Gap is known from are Scotland, the Balagan Formation, that's supposed to be extending for most of Romer's Gap, and then Nova Scotia and New Brunswick, which are age equivalent, or generally age equivalent. And the new site that we basically want to put into context is the New Brunswick one. What's also really important for this talk is that the regional zonation charts for palynology are actually based off of Horton Bluff, Nova Scotia. So where one of these major sites are. And this is done off of multiple years of data by multiple people. But this has been used, I would say, ubiquitously within Atlantic Canada since the 1990s. So what Horton Bluff is actually known for, though, is basically these large bones of stem tetrapods. And that tend to be disarticulated. We have teeth, hip bones, leg bones, plates. Unlike the ones that you can find in Scotland, the tetrapods seem to be a little bit smaller, but also more articulated. This is important because what we're seeing as far as footprints, as well as the size or morphology of the footprints, is that the skeletons that are being found in Scotland are seeming to be more similar to the footprints in New Brunswick, whereas the skeletons in Nova Scotia are these large, very, very robust skeletons. So it's interesting to find out and figure out where we are basically in that Romer's Gap, but also basically where do we sit in that evolution. In addition to that gap in your invertebrate and vertebrate realm, we also have basically a gap in the plants. If we look at this graph, we can see that seed ferns, basically your conifers, ferns, and lycopods, are really not very diverse in the early Carboniferous. And part of that is because there's not a lot of places worldwide we can actually find this stuff. And where we do, it's completely different and at a family level different. So plants are rare and they tend not to be articulated. But what we've been finding in New Brunswick, it's not quite the same. So we've been finding the trees preserved in ecological context in abundance. What you see here are lycopods that are being found, and these are rather large, especially, and quite diverse and distributed. So this is an example of one rock face we have, and each one of these starbursts that you see is a lycopod, and basically being preserved tilted up. So this is the bottom, and basically you're seeing the roots of these lycopods. We also have lycopods that have really nice pinnules, basically preserved as well. We have different types of ferns, as well as we have seeds and sporangia. So the likelihood of us being able to actually extract some palynology, or basically using the palynology to hide back to the plants, is quite high, or potential is quite high as well. As we have a new family of fossil tree. And this is something that Matt and I've been working on for a while and impressed with other colleagues, is this basically, we think it's a cladoxylon, but what's really interesting about it is that it's articulated, that we have the branches attached to the tree, the tree trunk, as well as pinnules and different things. But this is massive. Just to give you an understanding of scale, the branches on this extend about 1.5 to 1.75 meters. So this is huge. We also have another type of plant that's kind of interesting that we're not really sure what it is. It looks like a lycopsid, but it's much larger in the cushions themselves. So again, this is all new and wonderful things, but they're also new to the Mississippian in general. But that doesn't really help us when it comes to when we look at the age dating. We don't have that much. But what's interesting is that we do have the spores of these. And understanding what we have for plants does tell us something about what we potentially could have for spores of the area as well. But we also do have to separate them from the plants because we don't always know the affinities. With that though, these are still considered big fossils if you think of macrofossils. Plants are still macrofossils. So with that, we have big gaps. Whereas with microfossils like palynology, we don't. That being said, we do still have some biostratigraphic challenges in the Tornesian Atlantic Canada. We don't have those lava flows or tuffs that I was talking about earlier. Basically, the way that we can get radiometric dating. And what we have around it, or any types of ones that we do have, there's very poor palynology because it's probably cooked just from the heat of the lava flows. We also lack the marine biostrat, which means that we don't have anything from the oceans of the time that we can compare to readily because these are probably more inland environments. So think of comparing to the oceans when you're in the Rocky Mountains. It just doesn't connect in the same way. Macroplant taxa are also inconsistent or lack enough data, and that's in need of revision holistically anyway. So palynology is the most consistent biostratigraphy we can use in this region to really tie to other places in Atlantic Canada, but also more globally to Scotland and otherwise. So the study area, or my study area, is actually along number 1 highway in New Brunswick between St. John and Sussex, New Brunswick, over here, and it also sits within Stonehammer Geopark. It's part of the larger Maritimes basin and part of the Moncton subbasin. The age of the rocks that are exposed are about 360 million years ago, again, are of the early Tornesian and consist of the Horton group, which is comparable to what we see in Nova Scotia as well. But I'm specifically looking at the Albert Formation. The Moncton subbasin at the time would have been a series of lakes and rivers and deltas that are basically coming off these highlands of the New Brunswick platform and different uplifts, making these alluvial fans into these more interconnected lake areas, which have been much more luscious than we originally would have thought. Previous paleontology work that's been done, we have fish from the lake environments, we have trace fossils from both lake and onshore environments, as well as plant debris that's been previously discussed. So back to the study area, we have about 20 kilometers of highway. It's discontinuous strata of the Albert and Bloomfield Formation. And I must have had a duplicate thing here. Sorry about that. So we've been doing geological mapping along the number 1 highway. So this is just a geological map with the number 1 highway basically running through it as that green line just for orientation. And what's really important about this is that, so again, I'm just going to repeat this slide just because, again, sorry about that earlier, but we have these rocks that are exposed that are of the early Tornesian. What's really important is that these are of the Albert and Bloomfield Formation. So equivalent in New Brunswick would be the Horton group. But all these different formations are different lithologies, and they're representing different types of rocks. So basically, what are we seeing? And when you see these different types of things, they look very what we call layer cake, or one after another. But this is often an oversimplification of the stratigraphy in the area. And the zones, or basically subdivisions of these palynology assignments, have been used to define members and formations. So when we look at this, you can see that it's all these beautiful colors represent different formations, but realistically, formations grade into one another. So you can have a lake in the same area that you have, or in the same plane of existence that you have a forest in the same plane of existence, you have a deltaic system. And what you'll find is that if we apply something like this ornation chart, that you can see the age ranges of known fossil species can go through these different types of environments. So at the end of the day, what I'm trying to get at is that when you're looking at formations, or when we're looking at sampling for these, we cannot use lithology or environment to dictate time because they're time transgressive. So the same spores can be found in different environments, and using those small changes in evolution, basically, again, can subdivide time. So back to my study area. We've looked at geological maps, and we've done some, but there's structural complications in the area as well. We've used LiDAR data to basically strip away what we think of as far as the vegetation on top to show us what the layers of rock are bending and doing underneath. So this is a remote sensing technique that uses light to form pulsate layer and measure ranges, variable distance to the Earth. And what we found is that the rocks are folding and dipping, with our oldest rocks basically dipping towards the Bloomfield. So in 2018, we did field sites and basically started taking samples from along the #1 highway. And each site sample has stratigraphic column, but also palynology samples in context, which are really important. These are just some of the highway sites that we visited, just to give you an idea of what they look like, and also the variable orientations due to the structural complications of the sites. But then also, how do we do this? So once we're at that outcrop, for example, this is the Bloomfield outcrop along the #1 highway, we start basically subdividing these layers and start drafting these layers into different, based on their thickness, based on their color, and based on their sediment size, as well as what we're finding in them. For example, in this case, we have a fossil forest at the base. Then what we do is we start taking rock samples. So these samples, and especially for palynology, tend to come from these gray layers, and that's partially because that means that they're not oxidized. So sediments oxidize when they're exposed to air. So if you think of soils being brown, whereas river environments will be gray. We are looking for these river environments, especially because we're looking for spores dominantly. So palynology samples are ideally taken from these dark gray areas, and then they're used and they're logged basically in a sedimentary log. So we take these layers of rock and we basically put them in a bar graph to show us where we are in context. Also, just to understand where we are also in time, like we discussed earlier, layers of rock are basically deposited one on top of each other. So as you can see with a time arrow, as you go up in time, the layers of rock change, and we take these palynology samples in that context. So that's just one site of many sites we did in the Bloomfield area. And then we actually did more sites, not just within the Bloomfield area, but elsewhere. And this is kind of a more complete idea of what we have as far as the Bloomfield transition into, Albert Formation transition into the Bloomfield of New Brunswick. So again, when we're taking these samples, we're mostly taking them from mudstones. But I guess the question is, how do we take these samples and eventually get the palynology out of it? We do have to clean them, and then we start sending them off for processing. So this is where the rock is degraded and basically put into an acid bath. There's density separation and oxidization because these have gone under a lot of pressure and temperature over the years. So we do need them a little bit lighter than they would naturally be. And what we end up is with a slide that, like you see on the right, that's sent back. And you can see some of that basically almost looks like pepper on it. Those are the palynology, or that is the palynology that I'll be looking at. So in this case, you don't just do palynology off of one, you do it off of series of slides over a section. And those will basically give us occurrence, and we can start telling basically what we're looking at. So we use a transmitted light microscope, like seen in the previous slide, to start looking and start identifying these based off of their morphology. And each one is unique and usually represent a single type of plant. So even if we don't know where the plant is, we do know that there are abundances of different types of plant at the time. Another really cool technique that we use is called scanning electron microscope. And this is where you put one of the spores into a vacuum and then send electrons at it. And we get this beautiful, basically detailed surface image of what these spores could look like. And this gives us a really good understanding of the morphology of the exterior. So when we're comparing to other spores and other sites, we can say, yes, this is really similar or not. We didn't do it for my project, but it would be very cool doing in the future. So with that, we start identifying these spores, and there's hundreds of taxa. So for my site, there's probably about 200 different types of spores. And if we compare that to the macro fossils in New Brunswick, we can see there's only about 13 plant species or plant taxa that we know of. So you can see the detail in the palynology is actually quite high in comparison to our macro fossils at the time. So once we've identified what we have, we start determining basically what trends do we see? What are the first occurrences, the last occurrences, abundances, and we use those trends to start subdividing in the time. So back to this very simplistic look at basically how a zonation chart would be made. You can see, like, for example, the trilobite has a limited time, but it does overlap with other different species of shells, for example. We have more detailed zonation charts of the Tornesian, and this is kind of what they look like. Just very simply, the very same thing. We have our first occurrences, we have our last occurrences, and abundances are marked by these darker areas as you go up in time, and then you have species basically as your x-axis. And you can see that the first occurrences and last occurrences can mark the beginning and end of certain zones. And that's how we age the rocks that I'm looking at. Much like I did for my project, where I took samples through a very specific section, and we can start looking at the first and last occurrences of those samples, we do have to compare them to something. Like I said earlier, Nova Scotia and specifically Horton Bluff I may not be the most appropriate word, but is more or less the type section for how we age date rocks of this time. So if we look at very specific and time relative species, we can actually start comparing what we have in New Brunswick to Nova Scotia. So what we have here on the left, we have the section, the members. All that really means is that this is what the rock is subdivided in as far as the lithologies. So these would be comparable to what I was talking about, the Albert and the Bloomfield Formation. And then we have C. distincta and S. cleviger, which are just really important species that are important to subdivide time in this. So basically, this is a transition zone that I'm specifically looking at. It's often referred to as the 2-3 boundary, or the Spleo-Trilete-Cavadii zone. So what's important, though, is that because this is the type area that we're comparing to and we're using zonation charts on, we want to find out what the context is. So for these two spores, this is how they present in my section. We have one example of cleavager, and then we have multiple examples of distincta through the whole section. So what we want to do is orientate these together. Because we don't have that many of the cleavager, we can orient it into this original section. So the Bloomfield strata is the highest part of the succession then. And if we approximate this to about scale as far as how much metered section or how much strata was logged, we can actually see that this is basically how New Brunswick would fit into our Nova Scotia equivalent. And what this is really looking at is that our tetrapod location, which comes from the lower portion of our section, we think might actually be older than the tetrapods that we see in Nova Scotia at Horton Bluff, which is a major site. So it tells us something about the age of these rocks, but also how they fit into the context of other sites. So in conclusion, palynology can be used to subdivide time and give us a better understanding of how similar sites compare locally and abroad, and give us age and context of the radiation of tetrapods into the terrestrial realms. So again, science never happens alone, and this is just a component of a much larger project and multiple different projects between Steve Hines, Adrian Park, myself, and Matt, as well as many co-authors. So thank you.